Greetings, everyone, and welcome to Human Biology, the online version of this class. So the way that this class is situated is that we're going to be exploring the human body as a specimen. And the way that we go through this exploration is that we're going to look at things from the outside in. So this first chapter is really going to focus on how the human organism fits into the grand scheme of life. Um, Every chapter will then go a little bit further deeper into the human organism until we get down into the actual organ systems themselves. So I've designed this class to really mimic a face-to-face anatomy and physiology class without quite the depth of A&P, but you'll still get a lot of really good information that you'll find useful to apply towards anatomy and physiology or any of the allied health programs. So without any further ado, let's go ahead and get started. So some of the things that you'll be learning from this chapter, and each one of the chapters and each one of the sets of notes has what we call these learning outcomes. So I won't take time for each lecture and discuss each one of these learning outcomes. Instead, what I'll do is I'll just have them posted and then you can always read them. The learning objectives and outcomes very closely mimic the homework assignments, the Learn Smart modules, the quizzes, and your exams. I don't give review sheets for your exams or study guides, but following these learning outcomes for every chapter that we cover will be an excellent way to prepare for your exams. So as I said before, we're going to kind of work from the outside in. We're going to look at how the human organism, the human, actually fits into the grand scheme of life. So we have these very different levels of biological organization, and we can start from the largest level, which is here the biosphere, and that includes everything that's on this planet, both the biotic and abiotic resources. And then we go down to the ecosystems. The ecosystems are going to be these very defined areas on the planet that make up the biosphere. Within each ecosystem are multiple different communities, and these communities consist of the plants and the animals in that area. Different ecosystems, an example would be like we have here a grassy savanna in Africa, or you could have the Arctic ecosystem, and within that Arctic ecosystem there are various different communities. Each community is made of a population. At this point here we start to get a little bit more specific because the population only consists of one particular type of species. So the community has multiple species interacting together, but at the population level we only have one specific species that we're focused on. So then when we move down to from population, those groups of species, then we look at the individual species that are there. Species are the organism, the individual tree, or the individual human in this case. And then we have the organism is made up of these multiple organ systems. For the human, there are 11 organ system totals that your book will cover. And then each of those organ systems is made of organs are made of tissues are made of cells are made of molecules, and molecules are made of atoms. So in order to do all the things that your body needs to do, it's going to require a lot of energy. And the way that we define energy is the capacity to do work. And the way that we have the energy to do the things we need are the same way that your car has the energy that it needs to do its job. And that means that it's going to require raw materials to be put into those engines, either your car or your body, in order for it to function properly. So for us, we humans, we are considered heterotrophs. So we have to consume other things in order to get our energy. So the food that we eat provides the nutrients, and those foods, those macromolecules, which we'll talk about in chapter 2, can get broken down into smaller monomers that we can use as building blocks to build up different molecules in our body. Now, this whole process of breaking things down, consuming food in order to get energy from it, that's really just called metabolism. So that's the sum total of all of the chemical reactions that happen in your bodies, specifically within your cells, that give you not only the building blocks or monomers that your body needs in order to do all the cool stuff that your body needs to do, but it also gives you the energy to build up these molecules in different ways. So we consider the sun the ultimate source of energy on the earth because remember I said before that we're heterotrophs, which means that we have to consume other things that have, in order to get our energy. Plants, on the other hand, are considered autotrophs. So they can make their own energy via sunlight. So whether you're an omnivore, a herbivore, or a carnivore, where we ultimately get all of our energy from is through the process of photosynthesis, because we have to rely on the plants that make their own food, and they do so via the energy from the sun and a very elaborate process we call photosynthesis. And so we either consume the plants or we consume something that did consume the plants there. So another concept that we're going to talk about, and a lot of this stuff here are going to be definitions, especially this first chapter, so that we can really understand these terms when we come back to them again in the previous chapter. So homeostasis is the maintenance of a constant internal environment. So things that need to be constantly maintained are your blood sugar level, your body temperature, pretty much all of your organ systems, the pH, or the amount of acids and bases in your blood. Pretty much all of your organ systems are going to require some sort of stability within that environment. If that stability is disrupted, then we can have all sorts of metabolic diseases, chronic inflammation, even cancers arise if we don't maintain this homeostasis. So homeostasis itself would be really hard to maintain without your body's ability to respond to a stimuli. So the body has an ability to recognize when it's kind of out of whack somewhat. So if your body temperature is either too high or too low, there's a special part in your brain that can detect that and then can lead a cascade of events that will get that body temperature back into the normal range. So as your notes say, the external stimuli, remove your hand from a hot stove, that is the internal stimuli. For an example, it's internal stimuli, adjust your blood pressure in response to values outside the normal. So kind of the same what we talked about with body temperature. Facts and things that tell us that an organism is living or differentiate something as living or not living, or what we consider the qualifications for life, are not only its ability to respond to the environment and maintain homeostasis, that very first one we talked about, but organisms also need to have the ability to pass their genes on to the next generation or reproduce. So we have to be careful when we use the terms growth and reproduce, because depending on what kind of species we're talking about, the growth and the increase in the number of cells, if we're talking about bacteria, means that the number of cells, the population is actually growing. If we're talking about growth in a human organism, we're talking about the overall size of that organism getting bigger. So if you think about yourself as a child going from elementary school to high school to now adult, or even from infancy to toddlerhood to school age children, there was a progressive increase in your body size. But growth can also mean an increase in the number of cells. So development changes that occur from fertilization to death. So you obviously do not have the same machine, same body plan that you had when you were an infant, and you may not have, depending on what your age is, the same body plan, or the things don't function in your body the same way they did 10 or 15 years ago. So living organisms also need, for that part that they need to reproduce, is that they have to pass on their genetic information to the next generation. And the way that we do it is that we use deoxyribonucleic acid, or DNA. So that's all the hereditary information that tells your cells what kind of proteins to make, what kind of lipids to make, how much of them to make, what kind of proteins to express, what it is that you phenotypically or physically should look like. That's all been dictated by your genes. Now, for every last one of us that are viewing this lecture, that we all have a series of mutations, many of which are silent mutations, which means that the physical appearance doesn't really change, because although the amino acid sequence is different, the physical appearance that came out is still virtually pretty much the same. So mutations are really simply just variations in the genes. And because we're not all clones of one another, we're all essentially mutants in one way or another. So mutations aren't always detrimental. And a lot of times when we think of mutations, we think about the bad parts of mutations. But mutations can actually be quite beneficial, and it is actually the basis for evolution. So speaking of evolution, what is evolution? Quite simply, evolution are a change in gene frequencies over a period of time within a population. Evolution happens on the population level. That's very important to remember, not on the organismal level. Because think about it. What sense does it make that you have evolved and now you have wings, but if you can't pass that trait on to the next generation because no one else in the population has that particular gene, and it's really really not very useful. So when we talk about evolution, we're talking about a change in gene frequencies over a period of time, typically a long period of time within the population. So when we refer to the term natural selection, that is the process by which evolution occurs. So that means that there are, as I said before, we're all mutants. So there may be some sort of new variation or new gene frequency that pops up here and there within the population. And because of that specific new variation that comes around, that may make that organism better suited for the environment than the old version of that. So because of that, this new species, since it's better suited for the environment, is going to be able to use more resources and have more offspring, so that in the subsequent generations, you're going to see more organisms that have this new variation. And that is what we consider natural selection, and that over time will be considered evolution. So adapting is over time populations that have more of this advantageous variation, as we talked about with natural selection, then they're more likely to use the resources and reproduce. So check your progress. I won't do these every single lecture, but just as the first one here, what are some of those basic characteristics of life? Need to be reproduced, respond to the environment, the different levels of biological organization going from the biosphere down to the atoms, and we can either subsequently go smaller or we can go larger. And the relationship between adaptations and evolutionary change, that adaptations are your ability to respond in an efficient manner to the changes in the environment, because environmental changes are going to take place. And then natural selection says it's going to select for those individuals that have the most advantageous traits for that particular environment. And over time, that population will evolve, because evolution happens on the level of the population. So how are we related to other animals? Well, if we look at the grand scheme of things, when we talk about living organisms that have those characteristics of life that we have agreed upon, we can take all of those living organisms and we can really group them into three domains: bacteria, archaea, and eukarya. The ones that human fits into is that we fit into the domain of eukarya. Archaea and bacteria are considered to be single cellular organisms for the most part, or they're prokaryotic cells, and they don't really have a nucleus, well as any of the organisms in the domain of eukarya, that we have membrane-bound organelles, including a nucleus on there. So while archaea and bacteria are both prokaryotic, interestingly enough, archaea are more closely resemble those of eukarya. So here we have that domain archaea, and if you look at the words, you can kind of see archaic or old in there. We believe them to be the oldest living organisms that we have on our planet. They are prokaryotic cells, which means they don't have a nucleus, very small. They can adapt to very extreme environments like hot springs. They can live in really hot temperatures. They can live in really salty places that most living organisms cannot live. So they're able to absorb or chemosynthesize food, which means that they can use chemicals in order to make their own food, kind of like photosynthesis, except that we aren't using light as the beginning source there. And it has very unique chemical characteristics. It's kind of set it apart from any other group of living things on the planet. Bacteria are also prokaryotic cells that have a various different shapes to them. They can be rod shaped that we call bacillus, or they can be cocci that we call that are ball shape. They can also adapt to different environments. There are even some bacteria that can use sulfur as their food source, or they can use petroleum as their food source and make energy out of that. They can go through photosynthesis, they can chemosynthesize food, and they also have an abundance of unique chemical characteristics. On to where we fit in the domain of eukarya, we have two major classes in that domain. So we have this old, what we call kingdom of protists, which would be algae, protozoan, slime molds, water molds, paramecium, that would be into the protist area. They are single cell, but they can be very complex. They have organelles, they have nuclei. Sometimes they can even be multicellular. They're able to absorb nutrients, or they can undergo photosynthesis, or they can even ingest food. So the picture that we have here is a paramecium. We then have the domain eukarya of the plants. So I shouldn't say two major kingdoms. There's kind of like a couple of different things that fit in here. We have plants, protists, and animals, and fungi that fit into this domain eukarya. So these are only two of those eukarya in there. So protists are eukarya, plants are eukarya as well. So plants, algae, mosses, ferns, anything that uses photosynthesis to make its own food. Then we have the domain of fungi. Those are molds, mushrooms, and yeasts. Some are single cellular, some are multicellular, like we have here in this club fungi. And then we have the domain, in the domain of eukarya, the animal kingdom. So things sponges, worms, insects, fishes, frogs. Notice here that sponges, worms, and insects are invertebrates. But most of the planet, when it talks about the animal kingdom, there are more species of sponges, worms, and insects than there are of any other vertebrate on the planet. So they make up like 90% of the animal kingdom. We're actually the minority in the animal kingdom. Multicellular with different specialized tissues that we're going to talk about in this course, and we have to ingest our food. So we're not able to just absorb food. We're not able to photosynthesize food. We have to actually ingest it in order to get energy. So as I said before, most organisms in the kingdom of Animalia, or in the animal kingdom, are inverts. So worms, insects, mollusks, sponges, and all that good stuff. Vertebrates, we have a nerve cord that's protected by a vertebral column. Fish, amphibians, birds, pretty much anything that has a backbone would be considered vertebrates. Vertebrates that have hair, fur, and mammary glands, such as ourselves, we would be considered mammals. So humans specifically, we are most closely related to, as far as other animals within the domain of eukarya in the animal kingdom, we're most closely related to apes. But things that distinguish us from apes are that we have a more developed brain, and it's been hypothesized that we have a more developed brain because of all of the fat that we are able to consume in the early stages of our evolution. We have more upright stance, more creative language skills. There are multiple different dialects of the same language. There are multiple different languages. There are multiple different inflections, tones, pitches. There's a very creative use of language. And then we're able to use a wide variety of tools. So we're not just taking a rock and then smashing it against something to open up a shell. We're able to use a very, very elaborate types of tools, and that's probably linked to, or it's been hypothesized, to the fact that we have opposable thumbs. So our thumb, if you take your thumb and you face it towards all your other fingers, it's opposite, or it's opposing all your other fingers. So having that opposable thumb allows us to do a lot of different things. We are members of the biosphere, which is kind of like duh. Um, and we are members of pretty much every ecosystem on the biosphere, except for some of those places we could find a way to live in them, but it'd be really, really uncomfortable. But that's something else that separates us from other animals, is that we can make temporary adaptations to a particular environment by just housing and heat and air conditioning that would allow us to live in places that most other animals wouldn't be able to stand. So this last little bit that we're going to talk about refers to the scientific method. And it's important to have an understanding of the scientific method, because there will be some research articles that we read throughout the course that you'll need to be able to determine whether or not this is a sound, factual, objective information. And just as a member of this beautiful planet, you should have some understanding and some ability to decipher truth from what is not truth, and science from what is not science. So science really is just Latin for to know. It's just a way of knowing about the world. So as a scientist, one should be objective, factual, and not subjective, which involves judgment. So it's very different from saying that you just don't like a certain phenomenon the way it is, and just say, well, it doesn't exist because I don't like it. It's not a very scientific thought. What is a scientific thought? It says this phenomenon may be happening, and here are the reasons or the evidence to support that this phenomenon is or isn't happening. So it doesn't have any judgment on it. It's not just because you don't like it, or doesn't agree with your value system, or doesn't agree with your personal beliefs. You as a scientist have to shelf your beliefs on a particular topic or subject and look at the actual factual data and information that's presented to you. In order to do this, it's important to use a process or a method. And the scientific method has been agreed upon for multiple scientists across hundreds of years to be the most effective way in order to determine the objective factual information to allow us to make these scientific theories and hypotheses. So it's really just a series of steps. So first off, you make some sort of observation. So you observe that there may be some changes or things that have happened. So what you want to do is that you then develop a hypothesis. So there's not just one hypothesis that you can develop. There are multiple hypotheses that you'll be able to develop, and you can always go back and change them and rework them until you're drawn to your conclusion. So you'll notice here that it's not just a single linear steps that we go through. There could be some looping back. So we have our hypothesis, and then we predict what should happen based upon our hypothesis. And then from that, we'll make experimentation or further observations. So depending on what our experiments and our further observations say, we can do one of two things. We can just predict our hypothesis and then go on to the next one, or we can we just project a hypothesis to be done, or we can go on to our remaining hypothesis and do the same thing over again. If that hypothesis doesn't work, then we go on to the third hypothesis and we make our predictions and we have experimentations. And if our experiments confirm our predictions, then we reach our conclusion. If the experiments don't confirm our conclusion, then we can go back and modify the hypothesis. So with the scientific method, it's really just an advanced form of troubleshooting. So your remote control for your Fire Stick isn't working. So the first thing that you ask yourself is, well, is the television or the Fire Stick plugged in? So you go, that was your hypothesis, that the Fire Stick isn't plugged in. So your prediction is that if I plug in the Fire Stick, then it should work. So you get up, you plug in your Fire Stick, that's your experiment, and the Fire Stick remote control still doesn't work. So you have to reject that hypothesis and you go to the next one. Well, perhaps it's the batteries that are dead. So your prediction is if I change the batteries in my Fire Stick remote, then I will get it to turn on and I can get to my home screen. You change the battery, set your experimentation. If that still doesn't happen, then you go back to another hypothesis. So you're constantly doing the scientific method. It's just that we call it shooting. So for any good experiment, we want to make sure that we use a deductive reasoning to test our hypothesis. So let's go back to the Fire Stick remote example. If I change the batteries in my Fire Stick, then it will work properly. So for this experimental design, and we're just using a really simple, straightforward kind of thing because we're looking at the Fire Stick remote, you're always going to have an experimental variable and a dependent variable, or the one that's going to be responded. Within that good experimental design, you also want to have a control group. So your test groups are exposed to the experimental variable. The control group is not. So the control group, you are controlling all the things about it and nothing is really changing. But the experimental group, the test group, that's the one that you're actually experimenting, this change in diet or this change in sleep or this change in whatever it is that you're trying to conduct your experiment on. A lot of times because humans, it's unethical to run experiments on humans and even some higher order mammals, we can use models, sometimes computer generated models, or we can even use lab rats. I know you've probably all heard of that, to control these different variables and have different groups. So as a part of the scientific method, once you have done all these experiments, you want to meticulously, which means very carefully, collect and analyze your data. So there are different statistical data software and tools that can be used to see what how likely you are to have made an error or how uncertain that value is. If we put this information out there, then we put it in a scientific peer-reviewed publication. Marie Claire is cool. Vogue is cool. 17 magazine is cool. Forbes magazine, really cool, but they're not peer-reviewed journals. What separates a peer-reviewed journal from just a normal popular publication is that before a study or information is put into that peer-reviewed journal, it has to be scrutinized by a large number of scientists, and they look at that information and they determine whether or not the likelihood or the validity of the statements made in that paper are accurate or not, or how likely they are to be accurate or not. So it's going to be highly scrutinized before it's publicized. With things that go into Forbes magazine or Men's Journal, those things just have to get through the editor. So the editor may or may not have any expertise on that particular article. If it's a good article, if it's a good piece, then they're likely to publish it. Articles that go into scientific journals, on the other hand, they're going to have to be scrutinized by scientists that are experts in the field in which that article was written, determine whether or not they will publish it. So when you develop a conclusion, you've analyzed the data, you've reached your conclusion. The conclusion of one experiment can lead to a hypothesis for another, and it just completely goes on. And so that's why we can use scientific information and studies for one particular thing, and it can be used to fuel research and understanding for other things. So a scientific theory is an accepted explanation for how the world works. So a good example of that is the cell theory. All organisms are made of cell. Now, for something to reach the law or a principle, that means it's accepted by an overwhelming majority of scientists. So a hypothesis is what we think is going to happen. It's a tentative explanation of how the world works. So you've probably learned it as a scientific or an educated guess, emphasis on the word educated. If a hypothesis is tested over and over and over again, and we're consistently getting the same results every time we test it, and no matter who tests it, it can be elevated to the level of a theory. Cell theory, all cells come from pre-existing cells. Law or principle is the highest level hypothesis can get, which means that it's accepted by an overwhelming majority of scientists, because every single time we look at it and everything that we do supports the way that this theory has worked so far. And so now we consider it a law or a principle. That is, evolution has been elevated to the level of a law or a principle because we can see it happening. Organisms, whether it's a plant, a bacteria, an animal, a protist, whatever, are constantly making these adaptations or changes in accordance with the environment based on the changes in allele frequencies. So here's a really good example of a controlled study. So we have the hypothesis that one antibiotic is better than the other one. The control group, with ulcers, they're going to get the sugar pill or the placebo. So remember, the control group doesn't have anything that happens to them. Then we have our experimental group that is going to get the new antibiotic. The two test groups receive one of the antibiotics. It's a double-blind study, which means the doctors nor the patients know which group they're in. So the ones getting the antibiotic, they don't know they're getting it, and the ones not getting it, they don't know they're getting it, and even the doctors and the nurses. So those double-blind studies allow us to not have bias when the doctors and the administrators of the test, they don't even know who's getting it. But when we say they don't know who's getting it, that there are, once we look through the patient's body, we can see metabolites or how their body has broken things down, whether it was sugar or was actually antibiotic. We can detect those markers in things like their urine and blood when we're trying to pull out our results in our conclusion. So in our conclusion, the antibiotic B had better results. So some challenges that are facing science today, and things that we use to face those challenges. Technology is the application of scientific knowledge of human entrance. Biodiversity is the number of relative abundance of species and the variation of genes within the ecosystems. Those organisms that have the most varied genes are more likely to go on and do well than those that do not. Those that do not have a lot of variability, they face the possibility of a much higher chance of extinction, which is a complete death of a species. Right now, biologists are personally alarmed by the current rate of extinction, and things that may be leading to this rate of extinction are things like climate change, emerging diseases, global warming, encroachment on different habitats or habitat destruction. So there are multiple different things that are allowing us to have this very high rate of organism extinction. And I want you all to be mindful of the fact that when we talk about organismal extinction, we're not just talking about the cute fluffy things like the dodo bird and dolphins and different types of mammals. We're talking about things like bacteria. We're talking about things like sponges. We're talking about species within the coral reefs because of global warming and climate change. We are having the pH rise because of the rising ocean temperatures. So the coral reefs are bleaching and they're dying out. We're talking about different plants, and it might seem at first blush like, why do we care about the coral reefs or the plants or the bacteria or the insects or the beetles that are becoming extinct? I don't like them anyway. But keep in mind that Taxol, one of the most potent drugs that we use for chemotherapy, actually comes from a tree in the Amazon, the yew tree. So you can imagine how many different natural resources that could possibly be going extinct that could be actually helpful to humans and to other organisms. So it is very important that we maintain as much of our ecosystem as possible, and not to do things to harm our natural inhabitant any more than we already have, but instead try to heal our planet, because everything that's on this beautiful globe is here to help us and can be here to help us and are here to help it as well. So we have emerging diseases are relatively new diseases. Some are new or increase in exposure. This comes from spreading out more easily or globalization. Some diseases change or mutate. Climate changes are changes in the climate or weather patterns as a result of human activity, specifically greenhouse gases like CO2 emissions and fluorocarbons and that kind of stuff. And global warming is an increase in temperature due to the increased carbon dioxide levels of the atmosphere, causing those greenhouse gases like CO2 to be trapped in here, and increasing the overall temperature of the globe. All right, so there is all of chapter 1, and I will see you again for chapter 2. Have a great day.